Viruses are everywhere. We've all had them, and in today's world, it's unfortunately very likely that we will get infected by one even if we haven't already. It stands to reason that we should understand the viral structure, so at least when we're talking about it, we understand the nature of the beast. All viruses have a genome that's either RNA or DNA confined by a capsid. A capsid is a protein shell, and if the capsid and the genome are very close or tied together, then it's called a nucleocapsid. Capsids and nucleocapsids are made of various protein molecules that form predictable shapes. Usually, they are arranged in icosahedral, which means 20 equilateral triangular sides, polyhedral, which usually means 6 or more sides, or helical, or spiral forms. Helical capsids, like the tobacco mosaic virus, or TMV for short, are thread-like or bar-like in appearance. Icosahedral capsids, such as those of adenoviruses, appear generally spherical. Their icosahedral geometry is seen only through an electron microscope. Most animal virus capsids are icosahedral. The icosahedron shell shape has the max internal bandwidth, if you will. It's the best shape with the most space. There are more than a few viruses that have an envelope of lipids, proteins, and glycoproteins that come from cells they've infected. This wrapping is actually called a viral envelope. Now, viral particles, also known as virions, aren't actually cells because they have no metabolic activity. They don't have a cytoplasm, nor can they reproduce, clone, or replicate without a host. Viral genomes are either DNA or RNA, linear or circular, and single or double-stranded. Approximately 70% of viruses have RNA genomes. These include, but are not limited to, the viruses that cause the flu, measles, and, you guessed it, COVID-19. RNA genomes can be either single or double-stranded and are typically linear. Retroviruses, like HIV, are RNA viruses that insert a copy of their genome into a host cell. They have a single-stranded RNA genome that is reverse transcribed into double-stranded DNA inside the host cell by an enzyme cleverly named reverse transcriptase. Other viruses, like those that can cause smallpox and herpes, have DNA genomes. DNA genomes, for the most part, are double-stranded and are typically copied in the nucleus. So, now we know a little on what actually goes into the making of a virus. May the virus infections you get in your lifetime be brief and not a big deal.